Okay, let's start the class. Up till now, what we have discussed in this chapter is, we have talked about the cell cycle, the definition of a cell cycle, and what we said is the cell cycle is divided broadly into two phases, that is the I phase and the M phase. Talking about the I phase, as we have already mentioned, it is that phase where various types of metabolic activities takes place, right. Next is, it is also called as the preparatory phase. We have talked about the different phases of I phase as far as G1, S and G2 is concerned. We have talked about that. What is so very important about that phase is, once out of all the things that happen, the most important thing that we have to say is always about DNA replication that occurs in the S phase of I phase. Hetu sobotse important point of the I phase, right. As far as the M phase is concerned, M phase ki hobo pare, either it could be mitosis or it could be meiosis, depending upon which cell we are talking about. If you are talking about a body cell, then obviously it will undergo mitosis, or jodi reproductive cell kotha ko, then it will undergo meiosis. Similarity between both the thing is that the parent cell will divide and ultimately will form daughter cells. In case of mitosis, the number of daughter cells that would be formed is 2. In case of meiosis, the number of daughter cells that will be formed is 4. As far as meiosis is concerned, it is also called as reductional division because there would be reduction in the number of chromosomes, and mitosis is also called as equational division because the chromosome number in both the parent cell and the daughter cell remains the same. Last class, I think we have completed the mitosis portion. Right. What we remember is, whenever we talk about mitosis, it has, the entire process has been divided into 4 stages, that is prophase, metaphase, anaphase and telophase. And out of all the points that we have written against each and every stage, there are few points in general that we have to remember. Whenever we talk about prophase, kiki monta se, condensation monta se, dehydration monta se. Next is spiralization of chromosomes, we remember, nuclear membrane, nucleolus, all the cell organelles ki hoy, gradually disappear hoy, centrioles start moving to the 2 opposite poles, spindle fibers start appearing, etc. etc. Ekini asile prophase or important point. Talking about metaphase, just one line, that all the chromosomes start getting aligned itself in the equatorial plane. Next, moving towards anaphase, this is the time when the centromere splits, and if the centromere splits, each and every chromosome divides itself into 2 chromatids, and on the other hand, what we can say that this is the phase where the chromosome replicates. Right, actually ki form kore, duta chromatid form kore, kintu duita chromatid ok ami ki boli kobo paru, chromosomes boli kobo paru. Since the number increases, so we say that the chromosomes has replicated or duplicated itself, right. DNA duplication, chromosome duplication, duita beleg phase. Right. Next, after that, we have discussed about the last phase, that is telophase. What happens in telophase? Opposite of prophase, right. At the end of telophase, we will see a cell. Hey, cell to ki thakibo, duta nuclei thakibo, and plus the cytoplasm starts to what? Separate, or the process of cytokinesis gradually starts. So I think today we will be starting the class with cytokinesis. So heading koriba cytokinesis. And cytokinesis also okoman last class of explain korisu. Again, likhi lua. just a rough diagram so that you understand. Diagram to pisot likhiba ha. Ekinto buzi paisu, ekin last class to koraisu no. Oh, animal cell, ha, plant cell or itia like likha nai. Ekini pisot likhim. Animal cell or diagram or pisot etu likha. Etu part holo. Okay, so what are the points to be remembered from this portion is, in case of an animal cell, cytokinesis occurs by which method? Because we have two methods for a plant cell and an animal cell respectively. Animal cell or groove method or the furrow method, right? Talking about a groove or a furrow is, I think in the diagram it's clear what is a groove and what is a furrow. The next point is groove kune form kore, who will help to form that groove in the cell during the process of cytokinesis is certain types of proteins that becomes active at that time. Cell tour bhitorot already hebilak proteins asile. Right, it was already there, but they will come and arrange themselves in the mid portion of the cell only during the process of cytokinesis, right. So, there will be certain contractile proteins that will help to form the groove. Another point that is important is the word called as centripetal order. We will be comparing the cytokinesis of the plant cell and the animal cell, right. Plant cell or keneke hoy, animal cell or keneke hoy, compare korim. Compare korile, the first point of comparison is obviously the method. One is by cell plate method and the other is by groove method. The other point of comparison is centripetal order ne centrifugal order. I think we understand both the meanings, no? Centripetal hole ki hobo, towards the center, or centrifugal hole ki hobo, away from the center. If you have noticed in the diagram, that groove to keneke form korse, towards the center, right. Groove to away from the center form korna, away from the center hole to groove to belek hoy jabo, right. So, the groove that is formed is always towards the center, and so in case of an animal cell, cytokinesis occurs in which order? It always occurs in a centripetal order. Is point clear? Okay. Next we will be writing about this one. Plant cell ki likhisila by cell plate method likhisila no, tar tolot diagram to akiba. Sorry. write a word that is called as phlegmoplast, right? Vesicles arrange kori pelai junto portion form kore, that is what is called as a cell plate. They have another name that is called as the phlegmoplast, right? So that arrangement ultimately it is called as phlegmoplast. We will be writing it down kintu prothome rakhi lua. So below the diagram ekhine likhiba. Is the process clear? Key important, cell plate method. Here what to? Second key important, centrifugal. Who forms the cell plate? Phlegmoplast. What is phlegmoplast? Collection of the vesicles. Who form the vesicles? Golgi bodies forms the vesicles. We have already discussed in the chapter cell, right? Vesicles boli kole, it is not only the Golgi bodies, but if you remember, endoplasmic reticulum also produce vesicles. One does parts of our endoplasmic reticulum, we had vesicles in that part also. Even in Golgi bodies, we have seen the formation of vesicles, right? But mainly formation of vesicles, if the question is asked like that, the credit goes to Golgi bodies. I think you will remember the two terms, no? Cis and trans, right? The cis phase and the trans phase. So ultimately, Golgi bodies or credit to jai vesicles kune form kore, Golgi bodies form kore, right? And kuntu side opera form kore boli question to ahile, it is always from the trans phase or the maturing phase that the vesicles are secreted. Okay. So this is all about cytoplasm. Once we have completed this, it comes to an end of mitosis. Mitosis we have discussed. After mitosis, at the beginning or together with telophase ki hobo cytokinesis hobo, and this is how the cell will divide into two daughter cells. So after this we need to discuss meiosis, but before discussing meiosis, certain points. So heading koriba points boli. এইটো হ'ল নেকি সেইখিনি হ'ল ওকে colchicine আগতে শুনিছা effect of colchicine dropper batch শুনিব লাগে অকমান ওকে So we are talking about certain points. So we have something called as mitotic poison. Mitotic poison ki hoy, nothing but certain types of chemicals or substances, and what would they do? They will inhibit the process of mitosis, right? As far as the various substances are concerned, yar bhitor question ahise regarding mustard gas. Colchicine is very important. Later also in your class 12 portion we will be talking about it, right? Ploidy jeti ami porhim, no? Tissue culture and all those. We have a chapter, no? Strategies for enhancement in food production or at a chapter ase. That I will be talking about all this. Question to keneke ahise, aru eta question ahise. Okay, even this one is important. Azide and cyanide. Sometimes the question might be asked, say, identify the mitotic poison, hetu to hudiboi pare. Second point hudiboi pare, say it inhibits which stage? It inhibits prophase. So, prophase word to monot rakhiba. Clumping of chromosomes kune cause kore? Mustard gas, etu porhiboi lagibo. Aru colchicine ki kore? So just read the line. It inhibits the formation of mitotic spindle. If the mitotic spindle doesn't get formed, then what will happen? Mitotic spindle form koribo lagibo na lage during the process of cell division. Mitotic spindle, or what is the work of a mitotic spindle? Mitotic spindle mane spindle fibers er jintu structure form kore na, tak ami mitotic spindle boli kom. That is a spindle that we can see in meiosis also, but naam tu ami mitotic spindle boli koi do. Oh yeah, obviously. So spindle to eba spindle fibers bilakor suppose important kaam ki? The chromosomes will like equally distribute koribo, right? Or what they will do is, if there is one chromosome, they will divide into two chromatids and the parts will move to the two opposite poles. If suppose the spindle doesn't get formed, then what will happen? Hey cell to hey chromosomes number kin teneke thakibo. If that cell goes for fusion with another cell, tetihale ki hobo? Chromosomes kini increase hoi jabo na jai. Bujhi paisa ne? The number of chromosomes will keep on increasing if the spindle fiber doesn't get formed, because it will not divide the chromosome, right? So ultimately this is something that is used to enhance polyploidy. Polyploidy, I think we understand, increasing the sets of chromosome inside a cell, that is called as polyploid, right? Normally ki thake haploid or diploid thake. Diploid condition or ke jodi besi thake, 3 sets of chromosome, 4 sets of chromosome, tak ami ki boli kom polyploid. Generally this is done from horticulturists bilake birat, they go for conducting polyploidy in certain types of plants which has economic value, right? Kisuman plants or economic value thake. Hey, economic value, suppose if you take a sugarcane or stem is the fruit that we eat, right? Hey, stem to jodi dangor koribo paru, but stem to jodi kabar belegor logot we make a cross, and then actually this is something that's included in the chapter called, I mean in the point that is called as plant breeding. Ami jeta plant breeding porhim no, tete ekhini bostu clear ke porhim, right? Dutal plant cross kori notun bostu eta produce koribole, all these features eblakor karone we need certain types of chemicals, and one among them is colchicine. What is the question that is asked is colchicine or kam ki? Colchicine jodi apply kora tetihale ki hobo? The number of chromosomes will keep on increasing. Why it will keep on increasing? Because the spindle fiber doesn't get formed. If at a cell divide kori ase, during the phase of metaphase, if you apply colchicine, tetihale ki hobo? Spindle fiber forming nokoribo. Spindle fiber properly coat form kore, metaphase form kore, and anaphase they will start pulling it to the two opposite poles, right? So if you have to apply colchicine, so colchicine could be applied in a negative sense, it could be applied in a positive sense also. Horticulturists bilake to increase the ploidy of a cells of those plants that has economic value, te ami positive aspect ot ami colchicine apply korim, right? Increase chromosome number to increase korim. Increase korile maybe that species will get better. Right? We can always experiment, right? So in that case, ami ketia apply korim aru apply korile ki hoy? These are the 2 points that you have to know. First, you will apply it during the stage called as metaphase, and what will happen if you apply it? The spindle fiber will not be formed. What will happen if the spindle fiber doesn't form? The chromosome number will keep on increasing, and that is what is called as polyploidy, right? Iman details nalage, plant breeding or details korim. As of now, A2 word, bas. Ha. নহবই ন, এটা সেল ডিভাইডে নহব। স্পিন্ডল ফাইবাৰ যদি ফৰ্ম নকৰে তেতিয়াহ'লে সেলটো ডিভাইডে নকৰিব। সেল কেনেকৈ ডিভাইড কৰিব? কাৰণ নিউক্লিয়াছ যদি ডিভাইড নাই কৰা, নিউক্লিয়াছটোতো আৰু দুটা নিউক্লিয়ালে ডিভাইড নকৰে ন, গোটেইকেইটা ষ্টেজ হ'লেহে তোমাৰ নিউক্লিয়াছটো ডিভাইড কৰি পেলাই দুটা নিউক্লিয়াই ফৰ্ম কৰিব। ইফ ছাপ'জ ওৱান অফ দ্য ষ্টেজ গেটছ এৰেষ্টেড, দেন অবভিয়াছলি দ্য চেল উইল নট ডিভাইড, হয়। আৰু যদি সেলটো ডিভাইড নকৰে ক্ৰোমোজোম নাম্বাৰ ইনটেক্ট থাকিব তাৰ লগত যদি বেলেগ সেল এটাই ফিউজ কৰে ক্ৰোমোজোম নাম্বাৰ বেছি হৈ গ'ল নে নাই so that is what is called as polyploidy right mitosis meiotic meiosis লগত যিমানবোৰ ইভেন্টস হয় তাক আমি meiotic বুলি কওঁ meiosis যদি spindle fiber form হৈছে meiotic spindle right meiotic prophase mitotic prophase the prophase during mitosis the prophase during meiosis right mitosis লগত related যিমানবোৰ ইভেন্টস হয় তাত আমি mitotic word to use কৰো আৰু meiosis লগত যিমানবোৰ ইভেন্ট হয় তাক আমি meiotic use কৰো right okay then we'll write the second point right spindle মানে spindle fibers বিলাকে যোনটো structure form কৰিব তাক আমি কি বুলি কম এনেকুৱা এটা structure form কৰে নকৰে right এটোক আমি কি বুলি কওঁ spindle so we have to talk about the types of spindle two terms regarding spindle right or mitotic spindle or spindle fibers বুলি use কৰো spindle বুলি কওঁ mitotic spindle could be of two types right spindle দুটা টাইপৰ এটা হল amphiastral এটা হল anastral this word anastral or amphiastral remember the word called as astral rays আমি কৈছিলো no we have talked about astral rays কি হয় if you are talking about this as a centriole তাৰ পৰা সৰু সৰু microtubules কিছুমান ওলাই তাক আমি কি বুলি কওঁ astral rays right astral rays or পৰা astral word to আহিছে amphiastral what we understand by the word amphi both right so yate amphiastral mane ki we are talking about the presence of two ester in a cell normally আমি যোনটো সেল ডিভিশন দেখা পাও তাত সেন্ট্রিওল হে সেলটো সেন্ট্রিওল কেটা থাকে দুটা রাইট দুটা যদি সেন্ট্রিওল থাকে অ্যাস্ট্রাল রেজ কোনে ফর্ম করে দুটা পার্টত ফর্ম নহব জানো right if you talk about two centrioles moving to the two opposite poles then both the centriole will have what astral rays so if you see both then obviously we'll be using what amphi so amphiastral spindle formation ami code dekha pao in case of an animal cell in case of a plant cell astral rays present or absent obviously absent kyo absent karon centriole absent plant cell ot centriole thake jano plant cell ot centriole na thake so plant cell ot jedi centriole na thake centriolor pora he astral rays bilak develop kore so jedi na thake so what will be using a word that is called as anastral now another point that you have to know is no doubt centrioles are absent in a plant cell that does not mean spindle fibers does not form in a plant cell animal cell to spindle fiber form kore ba mitotic spindle form kore plant cell to mitotic spindle form kore animal cell mitotic spindle to kune form kore the centriole in case of a plant cell the centriole is absent does that mean spindle doesn't get formed no spindle gets formed centriole নাই কিন্তু centriolor যেবিলাক proteins দরকার to form that spindle gets synthesized in the plant cell during the process of cell division they don't have a proper structure from where the proteins will synthesize and ultimately they will form the spindle fibers during the process of cell division but at the time of division but jetia spindle fiber form kora time ahe tetia plant cell to ki kore they will synthesize those proteins that are essential for the formation of the spindle fibers spindle proteins is the point understood so hetu point morat rakhiba centrioles absent that does not mean spindle absent spindle duita cell ote thakibo understood next third point So amphiestral or anestral about question ahise. So amphi in case of a plant cell, anestral in case of a amphi in case of an animal cell, anestral condition in case of a plant cell. So tak anestral spindle buli koy aru amphiestral spindle buli koy. Okay, whenever we talk about abnormal mitosis, as the name suggests, no, mitosis jigita step pori, so there will be slightly different. Certain extra things will occur that will make the mitosis process, something different from the normal that we have discussed, right? When we will start with this, do you remember a word that is called as denomitosis? Denomitosis monot ase niki? Moi khodisu kiyo karone, because this is a portion that we have done in the class. Okay, do you remember this word called as Mesokeratic nucleus? Mesokeratic nucleus. Nucleus buli kole kenikwa nucleus thake? Eukaryotic and prokaryotic. Nucleus kenikwa type? Duta type no? Eukaryotic or prokaryotic. So prokaryotic nucleus ki hoy, eukaryotic nucleus ki hoy, we understand. But we have also discussed about a word called as Mesokeratic nucleus. Kot korisile? Protist, dinoflagellates. Right? We have discussed a word called as meso, because the question that is asked is mesokeratic nucleus is a characteristic of, or it is seen in. Just one line that you have to remember. What is a mesokeratic nucleus? What are the changes that occurs? Iman important no hoy. Khali ei bostu to monot rakhibo lagbo je mesokeratic word to hunile, you have to remember that this is a condition that is seen in case of dinoflagellates, and what's a dinoflagellate is a protist. Right? So jedi mesokeratic nucleus monot jedi ase, then you will remember the process. Jedi monot nai tetial to monot nai aru tetial to upai nai, I have to write it down. Ase ne nai monot nai no? Okay, chalo likha. Adenomitosis নামটো কিহৰ কাৰণে আহিছে, কাৰণ তাত dinoflagellates বিলাকত দেখা পাম. What is a mesokeratic nucleus? A mesokeratic nucleus is a nucleus where you see all the stages of cell division within the nuclear membrane. What was the first sentence about prophase that we have learned? That during the phase of prophase, the nuclear membrane degenerates. Tetla spindle fiber code form koribo. Nucleus to bhitorot nuclear membrane khon nathakile. Obviously nuclear membrane nathakile, no, it breaks off at the first, and then after that only the spindle fibers gets formed, and then gradually the cell divides. But in case of mesokeratic nucleus, all the stages of cell division will occur within the nuclear membrane. Nucleus or bhitorote hobo. Jedi nucleus or bhitorot hoy, that type of a nucleus would be called as what? Mesokeratic nucleus. Is the point understood? Prokaryotic, we understood that you don't have a nuclear membrane. Eukaryotic, defined, well-defined nuclear membrane present. Mesokeratic, it has a nuclear membrane and all the stages occurs within the nuclear membrane, it does not degenerate, right? Where will you find a mesokeratic nucleus? In dinoflagellates, and that type of a mitosis, I said no, nuclear membrane thakibo, gutebur, jimanbur prophase type ba, condensation of chromosomes, splitting of centromeres, etc. within the nuclear membrane dekha pam, and that type of a mitosis would be called as what? Denomitosis. Clear? ইমান ডিটেইলছত পঢ়িব নালাগে what is xenomitosis কেতিয়াও নাহে খালি এই wordটো পঢ়িম right আৰু where will you see right okay what about the first line to কি লিখিছে চোৱাচোন কিবা এটা ইউনিক আছে নে তাতে is there a special point in the first line what is the special point possess condensed chromosome right even in non-dividing nuclei in normal cases that we have discussed non-dividing nucleus in what form do you see the genetic material non-dividing কি দেখা পোৱা chromosomes দেখা পোৱা নেকি if a cell is not dividing if a nucleus is not dividing nucleus always has genetic contents inside it right if you observe it under a microscope a cell that is not dividing or not preparing to divide in that case within the nucleus the genetic material would appear in the form of what fine threads and the thread is called as what chromatin right chromatin or ki form kore chromosomes keneke form kore condensation ore so condensation of the chromatin network ketia dekha paba in a dividing nuclei yate ki likhise so this is the unique point and so it is called as abnormal mitosis clear हाँ এইটো এক্সেপশ্যন সেইকাৰণে আমি মেছ'কেৰিটিক নিউক্লিয়াছটো কথা পঢ়োঁ প্ৰটিষ্টাৰ বাকীবিলাক যিমানবোৰ পঢ়িছে ডায়েটেমছ পঢ়িছোঁ ইউগ্লিনা পঢ়িছোঁ ৰাইট সেইবিলাক বেলেগ এইটো এটা এক্সেপশ্যন ৰাইট চ' এইটো তুমি নাইডাৰ তুমি প্ৰ'কেৰিটিক ক'ব পাৰিবা না তুমি ইউকেৰিটিক কৰিব পাৰিবা ৰাইট চ' মাজৰ ফেজটোক আমি মেছ'কেৰিটিক ক'ম Sorry. Again, the note you don't have to remember. Here are questions just from the point of understanding and knowing that abnormal mitosis also occurs, right? If abnormal mitosis is in the form of free nuclear division, তেতিয়াহলে আমি কি দেখা পাম? Multinucleated condition. Free nuclear division মানে? A nuclear division not followed by cytokinesis is called as free nuclear division because we know whenever a nucleus divides into two nuclei, after that or simultaneously cytokinesis hobo lagibo. Right? If cytokinesis does not occur after nuclear division, that is what is called as free nuclear division. Clear? Okay, so ekhini asile abnormal mitosis, point number 3. Now we have to talk about point number 4. If you want to study about the process of mitosis, কোনবিলাক cells আমি পঢ়িম? So cells divided into two types, either meristematic or permanent. Cells বিলাক দুটা টাইপে নহয় জানো? Either meristematic or permanent. What is a meristematic cell? A cell that keeps on dividing is a meristematic cell. What is a permanent cell? A cell that will not divide, that is called as a permanent cell in general, right? So if you want to study mitosis, if you want to see the events that occurs in mitosis, you cannot be choosing a permanent cell for your study. হয় না? তেতিয়া আমি কি study কৰিম? Obviously meristematic tissues. So the best cell to study mitosis is obviously meristematic tissues. And out of all the meristematic tissues, meristematic tissues to in any part of the plant থাকিব পাৰে. Not necessarily it's present only in the root tip or it's present only in the root. Any part of the plant body will have meristematic tissues. But what is the best part to be studied is always the root tip, and which root tip, to be more precise, is always onion root tip. So question 2, onion root tip আহিব পাৰে বা root tip বুলিও আহিব পাৰে. Not necessarily onion would be mentioned, right? So the best part to study mitosis is always a root tip. Why a root tip? Because if you remember, we have talked about the zonation of a root. Morphology zonation of a root. So in other parts of a plant, for example, if you are talking about a stem or you are talking about a leaf, then a specific meristematic zone you don't see. Specific meristematic zone ami ko dekha so in the root. So it has a precise what meristematic area. So that is the best area to study the mitosis. So root tip is the best plant part to study cell division. Yeah. We have certain substances, certain chemicals, certain hormones, which one applied increases the process of mitosis or increases the process of cell division, right? So those substances that induces mitosis mane ki helps in frequent cell division or helps in faster cell division. Mitosis mane ki cell division nohoy jano. So those substances that will help in frequent or faster or cell division, those are called as ki mitogenes. Why mitosis? Myogenes to hobo pare no? Kila mitogenes, mitotic spindle, meiotic spindle to hobo pare. Why is it like this that we always use the word mitosis and very less we use the word meiosis? Because in majority of the cells, be like ki body cells, no? Body cells are ki hoy mitosis hai hoy. It's only on the reproductive cells that we see meiosis, right? So the most common thing whenever we talk about cell division is always mitosis, and that's the reason why we are using such terms. So mitogenes boli kole the substances, the chemicals, the hormones which induces mitosis. Mitosis induce korile ki hobo cell to divide kori goi thakiba and the part will become big. Okay, now from those who have heard something about hormones, have you heard a word called as plant hormones? Plant hormones. So porisa kunubai. So plant hormones boli kole, how many types of plant hormones we have to study? Okay, 4 types, 5 types, GNUPA. Okay, JD, if you have an idea about that plant hormones, name a hormone that helps in a cell division. Hormones ki ase plantor? Auxin, gibberellin, cytokinin, abscisic acid, ethylene. These are the 5 types, no? Tate kisumanok ami ki kon? Growth promoting hormones, because they will always have a positive impact. Some of them have a negative impact, that is called as inhibitory. For example, the best example is abscisic acid. And if you are talking about ethylene, both positive and negative. So one hormone that helps in the cell division. Auxin boli kole, okay, auxineo help kore, kintu to be more precise, cytokinin helps in cytokinesis. So cell division kune basically help kore, it's always cytokinin. Auxineo kore, auxin, gibberellin sobe koribo, because auxin dile ki hobo, a part will elongate, right? Kuntu part, root tip or the shoot tip, right? right? If you are talking about gibberellin, gibberellin ki kore, internodal elongation kore, right? We will be studying about all this later, right? So generally we are talking about kenuka dhoroner substances, the hormones, right? Auxin, cytokinin, gibberellin এবোৰ কি হয় plant hormones. Right? So these were the extra points. Next, likhiba significance of mitosis. What is the most significant feature of mitosis? Mitosis process to important hoy, kya important hoy? Baki points bilak jodi if you don't study also, one thing that we know, no, hotate injury hole ki hobo? Loss of cells hobo, loss of cells kini repair kune koribo? Cell division e, kuntu cell division? Obviously mitosis, right? So there are certain points because of which mitosis is important. Tar bhitor survival of the species also. Survival of the species ought, mitosis plays a major role. Kiho karne? Karon the chromosome number remains the same. Hoy ne? Nohole chromosome number jodi change hoy jay, species to change hoy jabo. Right? Okay. This is just a note. Any point that you did not understand, please ask. So, this is all about mitosis. Significance of mitosis, if suppose from MCQ point of view, though, you do not have to by heart, just at a point monot rakhile ho gol, right. From Board's point of view, if at all it is descriptive, then only you will be studying each and every point. If it is objective, no point of studying manke, no, okol khali just ever reading korile ami janim, right. Karon objective hoar loge loge, the pattern of NEET and the boards is almost the same, right. So, like NEETor karone jeneke porhim, boardsor karone ami tenekei porhim. So, is mitosis clear? What are the points that you will study? The tick points. How will you make the short note? The points that I have ticked, just write it down in a separate paper and the mitosis is complete, right. Iman khini pori thakhibo nalage, right. Is it over? Jodi hoy, next topic likhiba meiosis. Finished? Healing of wounds. So, hold on, okay. Huh? Last point to ki asile? Replace. Healing, we are talking about healing. Tamane tumi jeta tumar jodi injury hoy, cells kin ta okman kati gol. Cells kini kati joa mane ki cells kini mori joa hoy nohoy. Tate notun cells ako develop kore nokore. To notun cells develop keneke koribo? Kiba eta to hobo lagibo na, because if there is certain cells that have died and new cells needs to develop, we have already read cell theory. Cell theory ki koi, new cells to teneke form nokore, it always occurs from what? Pre-existing cells. So, pre-existing cells bilake ki koribo? They will divide. So, divide kori pele, they will do the process of healing. So, kuntu cell division? Mitosis hobo, right? Okay. So, we will be talking about the next cell division, that is meiosis. What we will do is, at the first part of meiosis, we will be writing the note, introduction part 2, kuntu point important meiosis term to kune disile, what is the other name for meiosis, etc. etc. Hekini likhar pisot we will be talking about the process. Some things that we already know. First, whenever we talk about meiosis, it occurs in germ cells or reproductive cells. Next, it is also called as reduction division. Why is it called as reduction division? Because the chromosome number reduces to half. Parent or jiman asile, daughter cells are half hoy jabo. Third, meiosis completes itself in two phases, that is meiosis 1 and meiosis 2. Meiosis 1 is precisely called as reductional division because it is at this time when the, meiosis 1 is called as reduction division. reason we all know, the normal reason that we always talk about, no, ki hoy chromosome number, right? Aru meiosis 2 is called as equational division, right? If we are talking about equational division, then mitosis of to ami equational division e koisilu, right? So meiosis 2 is exactly the same as mitosis as far as the stages and the characteristics of each phase is concerned. Prophase of meiosis 2, mitosis of ji hoisile hetu meiosis to hobo, right? Apart from the chromosome number je hetu half hoi jabo, reduce hoi jabo, hekhini ami pisot pohim, right? Reduction division boli kiye koise karon chromosome number komibo. Chromosome number code komibo meiosis 1 and meiosis 2, that is the point that you have to, yeah. Obviously reduction meiosis 1 jehetu tate komibo, right? And I think I have already said that you have to make a comparison between the three words. If suppose I talk about anaphase, tetale anaphase, anaphase 1 or anaphase 2 compare kori poribo lagibo. Prophase boli kole prophase, prophase 1 and prophase 2, this is the way you will study. Out of all the phases, maximum question has been asked about anaphase itself, right? So anaphase bostu to pohit. Next, talking about meiosis, since we talk about meiosis 1 and meiosis 2, meiosis 1 or ki thakibo prophase, metaphase, anaphase, telophase, exactly the same phases would be seen in case of meiosis 2 also. But in case of meiosis 1, the prophase is much more longer as compared to meiosis 2. Prophase akko, the entire process of prophase has been divided into 5 stages. Because meiosis or kisuman very important things that occurs. I think you have heard something called as crossing over. Yes. Right? Duta chromosomes thakibo, chromosomes cross kori exchange of genetic material hobo, that is what is called as crossing over. Code hobo prophase. Prophase hobo boli kole no hobo which stage of prophase, because we know in meiosis 1 prophase is divided into 5 stages. So against each stage important points kini poribo laibo. Questions kene kya hai? Important points about each phase and diagrammatic based question. Diagram di dibo, you have to identify whether we are talking about prophase, metaphase, anaphase or telophase or which phase of prophase, right? Jodi crossing over dekhai diye, then we understand that we are talking of pectitin of prophase, right? Then ekami bostu to porhim. Okay, first up let's write the note. Likha, the term meiosis was given by এখিনি আমি জানোৱে তথাপি লিখিব লাগিব The last point defines the process properly. What is meiosis? Reduction division. Kot occur kore? Diploid cells. Diploid cells occur korile ki produce kore? Haploid cells. Right? Haploid cells jetia produce kore, in the process ki hoy? Exchange of genetic material hoy. So, hey karne what we have read, there is a recombination of hereditary materials. Crossing over hole ki hobo? Exchange eto hobo. Oh, meiosis or proper definition. Okay. Meiosis kot hoy? A point to sabadi. Eukaryotic cells. Right? And meiosis kot hobo? Diploid cells or diploid cells kot thake eukaryotic organisms. This line is important. Is the line understood? In the process of meiosis, juntu cell meiosis undergo koribo, that cell is called as what? The cell that will undergo meiosis is called as what? Meiocyte. We have written a term called as meiocyte, no? Juntu cell of meiosis hobo tak ami meiocyte kom, right? If meiocyte undergoes meiosis, hey meiocyte tue keita daughter cells form koribo? Saita. How many times cell division will occur? Is it in one cell division or is it in two cell division that 4 daughter cells will occur? It is 2 cell division. So, if duta cell division hoy, and we have already read something called as I phase, whenever a cell divides it has to prepare itself. So, each and every cell that will divide will go for I phase, right. Yate the unique point is cell division dubar hobo, kintu DNA duplication ebare hobo, it is in the S phase itself. Right. So, dubar DNA duplication no hoy. So, DNA duplication kebar hobo ebar, cell division kebar hobo dubar. right? তাৰমানে once the parent cell is there, hey parent cell দুটা form কৰিব, hey দুটা cell আকৌ divide কৰি পেলাই দুটা cell form কৰিব, but at that time that I phase will no longer continue because they are already in the halfway of division. আকৌ preparation phase ত নাযায়, right? Meiotic cell. Meiotic cell বুলি ক'ব অকমান problem কি হৈছে না, the result of meiosis is also a meiotic cell. কিন্তু তথাপি we can say a cell that will undergo meiosis is called as a meiotic cell or a meiocyte. Proper word to কিন্তু meiocyte, right? Okay. If it's over, please say. Next, এখানে যদি হল এখন চার্ট লেখা, ইয়াতে এটা বস্তু লিখা আছে, এক সেকেন্ড। just to show that there are two cytokinesis in the process, cytokinesis 1 and cytokinesis 2, karyokinesis 1 and karyokinesis 2. So, when we talk about meiosis, meiosis 1, meiosis 2, since one cell will produce 4 daughter cells, so we have 2 cytokinesis, cytokinesis 1 and cytokinesis 2. I think the chart is clear, right? Meiosis 1 or ki thakibo karyokinesis cytokinesis, meiosis 2 to ki thakibo karyokinesis cytokinesis, because saita cell produce koribo lagibo. After cytokinesis 1 and just the beginning of meiosis 2, we have a resting phase, and that is what is called as interkinesis. Don't confuse yourself between the word that we have studied, interphase, or etia ami ki poriso interkinesis. At the starting point, আমি কি পাইছিলো যোনটো preparatory phase asile tat ki asile interphase asile aru etu ki interkinesis. One major difference between both the phases, that point that we have marked, DNA replication. Interphase ot ki hoy DNA replication hoy, interkinesis ot ki DNA replication no hoy. So that is the reason why we have written that statement before, no? In meiosis DNA duplication occurs only once. Ketia hobo hey preparation phase ot kite, right? After this, what we need to study is each and every stage precisely, aru ki events hoy, how does the cell divide, we have to talk about. Ekhon likhi loa, then we will be starting with prophase. Na tar agote points ketaman likhi lo. If this is over, points puli likhiba. Chart kon over no, each side. meiosis kot hoy germ cells hot no or reproductive cells hot. Again in case of plants and animals we have the two terms. Germ cells fully generally we don't use the word in case of plant cell. In case of plant cell ami ki kon sporogenous cell. And whenever we use the word sporogenous cell, it's something related with the sporophyte. What would be the ploidy twice n, right? So cell twice n holei to meiosis hobo. So plant ot meiosis kot hoy sporogenous cells bilakot meiosis hoy, right? Sporogenous cells bilake meiosis undergo. If you remember, we have talked about that archisporial layer koisilu no, pollen grain to keneke form kore. Okay, first year of koa nai. Okay, etu word to jano plant kingdom jehetu korisu, so we know the word sporogenous cells. So in case of plants, meiosis occurs in sporogenous cells. In case of animals, it occurs in germinal cells or germ cells, right? To be more precise. Understood? Ekoto nubuza nai? So these are some other points. After this, we will be discussing the process of meiosis, heading koriba process of meiosis. I can finish koriba. If it's over, please say. Microspore, megaspore, ebutu already we have studied in plant kingdom. Meiocyte or ploidy twice it. যোনটো সেলত মিওসিস হ'ব তাক আমি মিওসাইট বুলি কওঁ তো কোনটো সেলত মিওসিস হয় টুৱাইস এ নট এটো হয় ডিপ্লয়েড সেলছত হ'ব সো মিওসাইট ওকে আই থিংক ইটস ওভার okay now দুটা ওয়ার্ড এটা হল হেটেৰোটাইপিক ডিভিজন এটা হল হোমোটাইপিক ডিভিজন মিওসিস 1 হেটেৰোটাইপিক বুলি কয় মিওসিস 2 হোমোটাইপিক বুলি কয় So, the cell that will undergo meiosis has already covered which phase? The preparatory phase, right? Jikono cell eta divide kora agote, it has to undergo the I phase. So, that is the reason why we have written it. Meiosis hobo, war agote ki hoisile, preparation already hoye goisile. If you talk about preparation, the cell has already completed the I phase of cell cycle. After completing the I phase, then only it will go to the M phase. What is meiosis? M phase. বুজি পোৱা নাই এটা সেল মিওসিস আন্ডারগো কৰিব ঠিক আছে মিওসিস যদি আন্ডারগো কৰে তাক আমি ডিভিশন ফেজ বা মিওসিস মাইটোটিক ফেজ বা মিওসিস ফেজ বা ডিভিশন ফেজ বুলি কম নে নকম এটা সেলে যদি ডিভাইড কৰে তেতিয়াল তাৰ সেল সাইকেলটো হ'ব লাগিব না লাগে তো সেল সাইকেল হ'ব লাগিলে তাৰ G1 S G2 হ'ব লাগিব না লাগে so you have a parent cell producing two cells এই দুটা যদি মিওসাইট হয় if they are meiocyte then ki hobo eto stage to already hoi goise no yale ahibo tapse yate thakibo a cell to or eto jodi confuse hoise you talk about a cell that was in G1 apra S hol S apra G2 hol G2 apra ekhini stage ot juntu cell ase now the cell will ki hobo divide right a cell to kami ki buli kom meiocyte if it undergoes meiosis clear hoise nai right বুজা নাই এটা সেলে যদি মিওসিস হ'ব বা এটা সেলে যদি মিওসিস দেখাবো সেই সেলটো মিওসিস মানে কি cell division মানে কি something changes in the genetic material and cytokinesis karyokinesis so if suppose we are talking about a cell and this cell is a meiocyte ta মানে কি যে এই সেলটোত মিওসিস হ'ব সেই কাৰণে তাক আমি কি বুলি কৈছো মিওসাইট বুলি কৈছো right এই সেলটো যদি মিওসাইট হয় তেলে হি ডিভাইড কৰিব নকৰে ডিভাইড কৰিব এটা সেলে ডিভাইড কৰা আগতে কি কৰিব লাগে প্ৰিপেয়াৰ কৰিব লাগে তো এটো সেলেও ডিভাইড কৰা আগতে এই স্টেজ কিটা হৈ গৈছে নাই হৈ গৈছে সো প্ৰিপাৰেচন ফেজ অফ সবতছে ইম্পৰ্টেন্ট ডিএনএ ডুপ্লিকেশন আছিল নাই আছিল সো হে লাইনটো পঢ়া এতিয়া process of meiosis occurs in a cell that has already completed the I phase hoy gol he karone to divide koribo I phase nohol to divide no kore no So junto cell e meiosis undergo koribo he cell to already prepared hoy gol mane ki hoy gol DNA duplication already hoy gol Anaphase meiocyte to swa eta cell asile cell to e tak prepare kori dangor hoy ji hobo lagisil hol eitu stage palehi I-phase complete hoar pisot he cell to ki hobo divide koribo either mitosis hobo নহলে meiosis hobo to eitu cellor ki hobo meiosis to M phase hoy noy ba division phase hoy noy to etia prophase metaphase anaphase telophase yat dekha pam ne napam pam so junto cellot ami prophase metaphase anaphase telophase dekha pam tar to already DNA duplication hoy goise ne nai ei karone he line to likhisu meiosis starts from which stage meiosis boli specifically jodi mention kori it starts from prophase it is well obvious that meiosis is a division phase division hobo lagile I phase hoboi lagibo tak aru likhibo na lage thik ase so meiosis always starts from prophase etu ta hol ekhine likha hol niki side it's over okay so we'll be talking about the prophase phase of meiosis prophase is something that we have talked about in mitosis also so there are some common characteristics that occur in prophase and also in prophase 1 in prophase whenever we use the word prophase nuclear membrane gusi jabo nucleolus gusi jabo everything will disappear right and the chromosomes will get scattered in the cell হেখিনি হল প্রফেসর ক্যারেক্টারিস্টিক ডিহাইড্রেশন হবো স্পাইরালাইজেশন হবো কয়েলিং হবো কন্ডেনসেশন হবো right as far as prophase of mitosis is concerned precisely we have studied what early prophase mid prophase and late prophase right so similarly in case of meiosis prophase boli kole there are certain things that is common but we know that prophase is divided into 5 phases in case of meiosis so each and every phase kiba no hoy kiba special characteristic eta hobo at the end of prophase ki hobo nuclear membrane disappears are you understanding I mean mitosis or jeneke early mitosis or nuclear membrane does not disappear it is in the late mitosis that the nuclear membrane disappears so there are certain stages that keeps on occurring similarly in case of this also there are certain stages there are certain characteristics that we see in the cell so as of now since we are talking about the first phase of prophase of eta cellot jeti ami prophase or kotha ko no so we have this as a nucleus prophase starting out nuclear membrane thakibo ne na thake thakibo right because this is a meiocyte that will undergo prophase so it will have a nuclear membrane and inside the nuclear membrane we'll see thread like structures and this is called as key chromatid right so in this case also leptonemma or leptotin in this case also we will see the genetic material in the form of chromatids chromosomes etialake ami dekha nai po aru napao gradually during the process of prophase we will see the chromosomes সবতছে আগতে কি দেখা পাম ক্ৰোমাটিন দেখা পাম ক্ৰোমাটিন ক্ৰোমোসোমছলে কনভাৰ্ট হ'বলৈ কি কৰিব লাগিব কনডেনসেচন হ'ব লাগিব সো প্ৰথমতে কনডেনসেচন হ'বনে লাহে লাহে কনডেনসেচন হ'ব right so at the starting point what we will see is we will see the genetic material in the form of thin threads and so the name suggests leptothin means thin threads right so that word is more than sufficient if you know the meaning then you understand the genetic material will appear in the form of a thin thread later or gradually these thin threads ki arambho koribo condensation arambho koribo a thread dale condense hoy pele ki form koro short right during the process of condensation another point that you have to remember is this thin thread or this chromatin does not undergo condensation all of it together keneke hoy in intervals condensation hobo etur ki hobo condensation ki hobo in intervals and if condensation occurs in intervals tetiahale etu ki hobo অকমান দূৰ থাকিলে কনডেনসেচন হল অকমান দূৰ থাকিলে কনডেনসেচন হল ক্লিয়াৰ all of a sudden condense হৈ সৰু হৈ না যায় লাহে লাহে condense হ'ব just think about a thread right you have a rope or you have a thread it's a long one তাক ছুটি কৰিব লাগিলে গাঁঠি মাৰিব লাগিব নালাগে so gutekhini gathi maribo para or horu horuke knots you can do right so what happens in leptothin is the genetic material or the chromatin undergoes condensation but in intervals as a result the genetic material looks like this is the line clear right beaded in appearance eblak beads or nisna dekhisan nai beads kiyo karone hoise karon condense korise gutei dalot condense korise ne ebar condense korise akoman rest loise akko condense korise akko rest loise so we are using the word called as intervals right because of this we see the genetic material in the form of beads and beaded appearance of genetic material is a characteristic of which phase it's a characteristic of leptothin clear so kisuman points পঢ়িব লাগিব first point আমি এটু পালো second point regarding leptothin we will talk about flower bouquet চিনি পাও no bouquet of flowers right so the genetic material or the chromatids would appear like a bouquet in this phase keneke so we are talking about prophase Prophase, the first phase is leptothin. Leptothin, we know the nuclear membrane is still there. Bhanga to nai? Bhangise niki? Nai bhanga. So, yar bitorot ki asile? Genetic material asile? Tar bitorot ki hoise? Condensation hoise. This genetic material or these chromatids remain attached with the nuclear membrane remember this word telomere ki asile the tip of the chromosome right so nucleus tur bhitorot ki thakibo chromosomes ba chromatin material thakibo this chromatin material will remain attached with the nuclear membrane and ultimately it will look like a bouquet and so we use the word called as bouquet stage bouquet nisna dekhisen nai etu hoy yate phul bilak lagi ase right it looks like a bouquet it looks like a collection and so it is called as a bouquet stage and how is the bouquet stage formed the bouquet stage is formed by the chromosome remaining attached with the nuclear membrane by their tip that is called as telomere clear yar bitor jumlak genetic material ase floating to kori thakibo noare na float kori thakibo niki float to kori nathake kobat to lagi thakibo so kot lagi thakibo nuclear membrane khonot lagi thakibo so if you see it clearly it looks like a bouquet and so we use a word called as bouquet stage so if you are talking about leptothin some of the points that are important nuclear membrane nucleolus all the organelles is intact first point second condensation starts but in intervals so we see a beaded formation third bouquet stage it's also called as bouquet stage why because the chromosomes remain attached with the nuclear membrane by their telomeres so these are the 3 points that you have to study right eta baki khini likho what is the spelling of bead? B-E-E-A-D or B-E-A-D? A-D, no? কি হয় no? For spelling. So, as I said, meiosis has or meiosis 1 or it is meiosis 2, we have the 4 phases, no? Prophase, metaphase, anaphase and telophase. What is the first line about prophase? The general things that we always talk about it. It occurs in prophase, prophase 1 and it also occurs in prophase 2. After prophase, what happens is metaphase. Either it is metaphase or it is metaphase 1 or it is metaphase 2. Characteristic key, ekei nohoy jano? In all the 3 cases, the chromosomes will align themselves in the equatorial plane. But what we are observing here, since prophase is of a longer duration in case of meiosis, we have different stages that is observed, or there are different characteristics in each stage. At the end of prophase, ki hobo? Nuclear membrane disappears, right? At the end of prophase, ki hobo? Metaphase hobo. Metaphase ki hobo? All arranged in the equatorial plane. Kintu prophase to jehetu dighol, beleg characteristics develop kore, beleg characteristics we need to study. Prophase in case of meiosis, first is leptotin. What occurs in, or what happens in leptonin? Because leptonin or pisor junto stage now, that is called as zygotin. So again the question will be asked, what occurs in zygotin? Something changes would occur obviously in the genetic material, right? Some changes would occur in the nucleus, right? So in prophase 1, in the first phase, what you need to remember, just 3 words. First, leptotin, as the name suggests, is thin threads. So what you will remember? In leptotin, the genetic material always appeared in the form of thin threads the first time. Second, condensation occurs, so we see beaded appearance of the genetic material, bead dekhapam. Third, bouquet stage. Question I have, bouquet stage boli kuntu stage ok koi? Leptotin is called as the bouquet stage. There is nothing much to remember. Clear? Oh, another question that could be asked is about this point. Does the nuclear membrane degenerates during leptotin? No. Kintu prophase ot ki janu ami nuclear membrane degenerate hoy, kintu prophase of first thote degenerate hoy janu no hoy. So the question is asked about this point. Clear? No doubt we are writing many sentences, but we have just 3 words to remember from this stage: thin threads, beads, bouquet stage, right? After this, next likha zygotin. When is your next class? 6:30. Etu class keta lage 5:45 na 6? So what do you want me to do? Continue or stop it? Stop it. Okay, chalo then. What we'll do is zygotin we'll talk about it in the next class. Kintu ekhin loge kiba doubts ase neki swa. हाँ हाँ वो डाउट है? हाँ क्या डाउट है बोलो? Nuclear membrane will disappear only at the end phase of prophase. Leptotin क्या है? First phase of prophase. So first starting में ही nuclear membrane disappear नहीं करता. It's only gradually during the process that the nuclear membrane will disappear. So nuclear membrane totally disappeared ami ko dekhapam. What is the last phase of prophase? Leptotin, zygotin, pakitin, diplotin and dikinesis. It is in the dikinesis stage that you will see that the nuclear membrane has totally disappeared. Leptotin, it's still there. Zygotin, it's still there. Pakitin, it's still there. Right? At the last phase of prophase, that is dikinesis, you will not see the nuclear membrane. Right? Or ask your doubts if you have to be clear. Karon iman simple ke questions nahe no, ghurai pokai dibo. You have to know the concept, you don't have to by heart, right? So just know the concept. Another thing that you will know or you'll remember when you study meiosis is, meiosis occurs in a germ cell. If you are talking about a reproductive cell, tar bhitor jiman khini chromosomes thakibo, that will be of one parent ne 2 parents. Are you understanding the question? Meiosis kot hobo? Germ cells were reproductive cells. Because of meiosis, what will be the product? Gametes form koribo. Or else in case of higher organisms or meiosis war karone ki form kore? Egg form kore, sperms form kore, right? So ultimately, juntu cellot meiosis hobo, hey meiosis wa cell tut, the genetic material that is present, tat ki thakibo? Already tumar pakitin stage or crossing over hoi goise nai. Characteristic of both the parents thakibo na thake. So do you understand something called as homologous chromosomes? Homologous chromosomes means if you are talking about a cell that will undergo meiosis, each and every genetic material, each and every chromosome will always be in the form of a pair. One chromosome from the father side, one chromosome from the mother side. Jihetu meiosis or kotha koise, if you are talking about mitosis, it's all the characteristics of a single parent. Meiosis, কি ফৰ্ম কৰিব এগ ফৰ্ম কৰিব বা টেষ্টিছ স্পাৰ্মছ ফৰ্ম কৰিব বা উই হেভ দ্য গ্যামেট ফৰ্মেশ্যন কি হ'ব ছেক্সুৱেল ৰিপ্ৰডাকশ্যনৰ কাৰণে ক্ৰছিং অভাৰ এক্সচেঞ্জ অফ জেনেটিক মেটেৰিয়েল হ'ব নহয় এক্সচেঞ্জ হ'ব লাগিলে দুটা কেৰেক্টাৰ থাকিব লাগিব নালাগে right so in the next class when we will be talking about the zygote phase we will be using a word called as homologous chromosome ঠিক আছে chromosome homologous chromosome heterologous chromosome এই word গিটা অকমান চাই আহি পাছো ঠিক আছে I think that's all for today thank you for the next class zygote লিখি থবা sit down